Okay, thanks everyone for coming to the Tech Talk today. Neil Miller is going to speak to us today. He's the Director of Informatics at Children's Mercy. He's going to talk about challenges with genomic medicine. So everyone, let's give Neil a warm welcome.
Thank you all. Thanks a lot for having me, Kevin and Kevin. It's a real opportunity, great opportunity for me to be here talking to you guys, and super strong engineering organization. I have to admit that I'm really used to giving science talks. It's been a really long time since I've actually given an engineering talk, so this is kind of a treat for me. So, as Kevin said, I'm the Director of Informatics for the Center for Pediatric Genomic Medicine at Children's Mercy. What that means is that I'm in charge of software development, IT, and bioinformatics analysis for a working genome center that's integrated with clinical practice at Children's Mercy Hospital. So I thought what I would do in this talk is tell you a little bit about some of the challenges that we face, some of the common analyses that we do, and how we're hopefully solving that in the informatics group. Just a tiny bit about myself. I entered the field of bioinformatics as a software engineer in 1999. It was completely by accident. I got hired by a biotech in Boston, basically because I knew Perl, and that turned out to be the language of bioinformatics. I'd been a software engineer for several years before that. And I found my way to the National Center for Genome Resources in Santa Fe, New Mexico, where I ended up working with my boss, Stephen Kingsmore. And he and I left in January 2011 to come to Children's Mercy to start the Genome Center, again with the goal of being a clinically integrated genome sequencing center. Currently I've got a small but mighty staff of informaticians working for me with a skill set ranging from software engineers to bioinformatics analysis to high performance computing engineering. Within the hospital, we've got partnerships with physicians in all the major areas of the hospital, clinical genetics and counseling to work with the patients, and of course, the collaboration with our Center for Bioethics. So our center's main goal, what we focused on is using genome sequencing to diagnose genetic disease. And one of the things we've been doing is an ultra-rapid whole genome sequence of critically ill kids in the neonatal ICU. And this is something that got us a fair amount of attention late 2012, put us on the front page of the New York Times even, and got us nominated to be one of the top 10 medical breakthroughs of 2012. The case for this, I'd like to walk you through a little bit. So every year, there's about 1,000 newborns who are admitted to CMH's NICU. Nationwide, 6 to 7% of all newborns end up being admitted to the ICU. It's believed that genetic disease is the #1 cause of infant death in the NICU. It's hard to know for sure because most of these kids don't get diagnosed. There's over 4,000 known genetic diseases. What that means is that for a physician with a sick kid, it's really difficult to actually make a diagnosis by looking at their symptoms. So this practice of what we call genomic neonatology, what that means is we're going to use a patient's genomic information not only to diagnose their disease, but to hopefully manage it, direct treatment. If you do clinical diagnosis, what you're doing is you're targeting symptoms and prescribing drugs based on that. If you have a molecular diagnosis, what you're ending up doing is actually targeting the disease mechanism. It's a lot more specific. This also allows us to look at what the patient's prognosis is going to be and predict complications, and basically counsel the parents maybe about their future risk for having more children. I'm going to walk you through a very quick vignette. This is a child that we saw last April. Child came in with acute liver failure. It was believed to be genetic. They could not figure out what was going on despite a lot of testing. So we entered it into our rapid protocol, start with giving genetic counseling and admit the family into our research study, get consent. We draw a sample, blood in the NICU, send the samples to our lab, purify the DNA from the blood, get in sequencing, and then we go through an ultra-rapid genome sequencing that takes 25 hours. At the very end of that is when it ends up in me and my team's lab. What we do is we then analyze all the data that comes off. We find the meaningful differences in the patient as compared to the human reference genome. We identify them, and we characterize them, and then we sift through them to try to see if we can find any that are explanative of the disease. So to start with, we ended up analyzing 2.8 billion nucleotides of the patient's genome. Of that, we found about 4.8 differences or variants. Of that, about 1 million of them were very rare. So the idea is that disease-causing mutations are going to be very rare in occurrence. They're not going to be very common within the population. So when you're looking for something that's going to be causative, you're going to be looking for things that are very rare. Then further, we have 900 that are rare and expected to be severe. And of that, looking at the patient's symptoms, we're able to figure out there's 340 diseases that are potentially relevant to the patient. And within those 341, we have 2 variants. And that leads us to a diagnosis. And further, for this particular case, we're able to change the treatment, the outcomes, the liver function of the child returns to normal, and the baby's now gone home. She's still sick. She's going to have this disease for life, but now it's managed. So a key part of this, how we're able to do this, is what's called next-generation sequencing, which I don't know if it's a term familiar to you guys or not, but basically it's a way of describing a new class of DNA sequencer that came out 2005 or so that really changed the landscape of sequencing. And when we say next gen, that means it's what's current. It's differentiating from the first gen, which is called Sanger sequencing. It's massively parallel, ultra-high throughput. What that means is rather than sequencing one piece of DNA at a time, it's sequencing millions or even billions right all at the same time. They're very short pieces of DNA that come out, short read lengths, and they're relatively high error rate. So remembering back that the first human reference genome was produced in 2003, it took 13 years for them to do that, and it was a cost of around $3 billion. 300 million of that was for sequencing alone, the rest was for informatics analysis. So by comparison, one of the instruments that we use today, the Illumina HiSeq 2500, is now capable of producing 1 trillion base pairs, which is the equivalent of 8 human genomes, and that's in 6 days for a cost of less than $3,000 a genome, just in sequencing reagents. This graph is basically traditional to show in any talk on next-gen, where you plot the increase of sequencing output against the decline of machine cost according to Moore's law. Basically, it's just to show you that the sequencers are very rapidly producing more data than our advances in compute hardware can keep up with. So this is leading to something that we're calling the data deluge. Informatics and the ability to analyze this stuff is the new bottleneck. The analyses that you end up doing on the short read data, the next-gen data, is very compute intensive, and the data that it produces are very large. So there's a distinct set of challenges that are, a lot of them are just engineering and IT challenges, and a lot of them are algorithmic and informatics challenges to dealing with this stuff. So just to remember, the human genome, it's about 3 billion base pairs. That's equal to, if you were to type 60 words a minute for 8 hours a day, you'd be doing that for 50 years to do one genome. Buried in there is 22,000 genes. A lot of times we look at that and think that's the most relevant part, especially to disease. It's 2% of the genome. It's 100,000 proteins that are produced. And humans differ from one another by about 0.04%. That's equals around 10 to 12 million base pairs that differentiate you from the person sitting next to you. And it's really those differences that we're interested in identifying when we're doing genome sequencing. Some of those are completely benign differences. They account for your height or your hair color, and some of them are disease-causing or lead you to predisposition for disease. So I thought it might be interesting to walk you through computationally how we end up doing this. So what I'm going to show you is called variant detection. So that's our word for these differences between each other. called variants, and we always describe variants in terms of a difference in an individual and the human reference genome sequence. So our machine, as I said, the sequencer produces 1 trillion base pairs, and it produces these in these very short pieces of 125 base pairs of C's, T's, G's, and A's. And so the first step is to align these or map them to where they came from on the genome. The second set is to find places where the reads differ from the reference. The third step is then to do what we call variant calling, which is basically looking at what base are the reads calling in consensus compared to the reference genome. In this case, you can see the position 129990. We have a change from A to a C that most of the reads in that position are calling. And then finally, we characterize that, knowing that there's a difference there. Can we tell anything about what it means? One of the things we do is look at external databases to see if we can match it up and see if anything else is known about it. In this case, that's what I'm showing you here is an accession number in a public database. So back to our medical genome sequence, our ultra-rapid, which we're calling STAT-seq. Knowing now a little bit about what the process is, I can show you what the timings are at different stages. So the very first step is to take the patient sample, turn it into DNA, and then get it onto the sequencer. So that takes around 30 hours with our new rapid machines. Then the process of sequence alignment takes about 15 hours on our compute cluster. A lot of the compute problems in bioinformatics tend to be what are called embarrassingly parallel problems, where it's not that we have big problems that the whole cluster works on together in a distributed way. It's really more like we've got lots of little problems, and we spew them out over the whole compute cluster and let everything run in parallel. That's what sequence alignment tends to be. Once we've done alignment, variant detection can be done in 3 hours. The characterization process takes about 1.5 hours, and then the interpretation means a person actually pulls up the results and looks at them, and in many cases we can do this in as short as a half an hour. That's the state of the art today. We're in the middle of doing some updates that are going to bring this down. So our sequencing partner Illumina is working on a new machine that's going to bring down the sequencing time to 21 hours. Alignment, they've produced optimized software that'll now do this in 1.5 hours on a single compute node as opposed to 15 on a very large cluster. And variant detection they can now do in about a half an hour. I think this is a common trend in engineering. Once you identify a problem, you're able to really focus on it and optimize it down, and that's what's happening with short read alignment and variant detection. So with those advances, we're going to be able to turn this into 24 hours, hopefully sometime this summer. So I mentioned the data are very large. Just to give you a sense of that, I went and looked at a case that we did just last week and wrote down how much disk space we were using up to store this genome sequence. So there's about 1.5 terabytes of primary data and 300 gigs or so of secondary data that we end up basically throwing away because it's not really useful and the costs of archiving it are just too much. But there's still a large pile of data that we keep permanently, and it really comes out to be about 177 gigabytes of permanent data for each human genome that we're keeping. Biologically, that's over 1 billion reads. It's covering each position in the genome 37 times. It's 4,900 potentially diagnostic variants. But from an IT perspective, it's 177 gigs. And with our new sequencers, this is just part of the output of our sequencing center right now. We're able to do 2 of those every 24 hours, or if we run a slightly different configuration, we're going to do 16 of those every 6 days. So what that means is just in whole genome sequence, which is just part of what our center's doing, we're producing 3 terabytes of permanent data every week. So it takes a lot of computers to deal with this. We have a primary compute cluster. It's 36 nodes in varying configurations of cores and RAM. But in the end, we have 1,200 core cluster with 6 terabytes of total RAM. We've got a reasonably sized database server, 96 gigs of RAM. But really, one of the most important things is our disk array. We've got an Isilon X400 storage cluster. I'm not sure if the audience is familiar with Isilon, but what they do is they produce this product that uses something called 1FS. What 1FS does is take several separate data storage nodes and allow them to present as a single file system. And what that means is that you have a very scalable, high-performance system that you can add to just by buying additional nodes without having to migrate data between systems. So we currently have around 800 terabytes of this usable. We're planning to grow over a petabyte in 2015. Usually at this point I say that that's more than the rest of the hospital combined, although when I say that, it doesn't include anything that Cerner's storing for us, so maybe that's not a good one for this audience. Our cluster has to accommodate a lot of different kinds of jobs. There's high memory jobs versus high compute jobs. And what that means is that in order to meet all the needs, we have it configured fairly flexibly rather than optimizing to any particular style of job. Here's a diagram of how we've got this network laid out. The whole Genome Center compute resources are living on their own subnet that's separate from the main hospital network. So folks out on the main network can see the primary storage system through CIFS, and they can access a variety of different applications running on the web server and the database server internally. Most of our compute is handled by SSH directly to the cluster head nodes, and then submitting jobs through the Oracle Grid Engine management system that then takes care of farming them out to the compute cluster. So you can see there's quite a collection of technologies at work. We've got applications in Ruby on Rails, Django, Java applications running on Tomcat. Back end is MySQL database, the Isilon system, of course, and then our compute cluster was built by Advanced Clustering Technologies, which is right here in Kansas City. All of these servers are running CentOS Linux. So as you might imagine, backup is a serious issue. When we have a primary data storage that's 800-something terabytes, and we're in total generating about 10 new terabytes a week. So what we've done is deployed a combination backup system that relies on the SGI Infinite Storage Gateway, which sits as a front end to the SpectraLogic T950 tape library. And so the tape library is 950 slots of LTO6 tape. It's 850 terabytes usable. We can expand that up to 2.5 petabytes. And then the SGI gateway is, it has a local cache system that it can keep a certain amount of data resident for fast access. Mostly what it's doing is taking charge of copying data from the primary system over to the backup system, and it's doing that twice, once so that we have online storage in case of a serious disaster, and then one copy that's going off-site for permanent archive. Within the network, security and privacy, again, is a serious issue. We get some benefit by being a separate network within the hospital network that's not visible to the internet. All the data is stored with de-identified accession numbers, where the link between what the accession number is and who the patient actually is localized in a place where hardly anybody can see it. Certainly where no one can see it by accident. We are encrypting some of our protected health information databases. We're encrypting those right on disk, and of course websites are accessed through SSL. We do a fair amount of custom software development within the center, and we're a small team, but we do try to adhere to industry best practices, and so I call it sometimes an informal CMMi level 2 process, although I should say that we haven't actually gone through CMMi certification. So, but basically the idea is that we've got build management, we've got versioning, everything at the end of the day, all analyses that we do need to be auditable. We need to be able to look at how we produced each result, what were the version numbers of every piece of software that was involved. We need to be able to replicate it if we can. I should say that's a challenge, actually. That was one of the biggest challenges of coming into the clinical environment, because from the research world, things are sort of fast and loose. And this is a dynamic field. Software that we're using is changing all the time. And so it was a challenge to come up with a set of processes that were going to let us be nimble, agile, if you will, and as well as still adhering to this need to be fully auditable down the line. Okay, so back to our problem of analyzing variants. So there's 3 different types of sequencing that we're doing most of the time here. The one we're mostly focusing on right now is whole genome, and so again, that's producing around 4 million variants per patient. The issue is finding the small handful of variants that are meaningful to the patient, and so to handle that, we've developed something that I like to affectionately call the Nordic Suite, which is 3 tools that we developed ourselves: STAGA, Runes, and VIKING. And what we found is that looking at the patient's data and taking it down to that small number really requires looking at both the patient's symptoms as well as the variants that are produced and integrating those together to come up with a tool that allows people to easily drill down to what's relevant. So STAGA is our tool that manages the patient's symptom terms. Runes is the tool that handles the characterization of individual variants. And then VIKING is a client tool that people use to merge those 2 data sets together. I'm going to use another patient case to illustrate how we use this stuff. So these were our very first 2 patients that we enrolled in our study in 2011. This was a pair of sisters who had ataxia. They were starting to lose control of their motor functions. And they had cerebellar atrophy. They were enrolled to study. They'd been on what commonly gets called the Diagnostic Odyssey. It'd been 5 years since they'd come to the hospital. They'd gone through $23,000 of testing and still no answer. So the first step in looking at their case was to go to our STAGA tool. And so STAGA is a lightweight web application. Current version's written in Ruby on Rails. We've got a Java version on the way. The idea is to put the symptom entry into the hands of the physician who's not a genetics expert. And what STAGA does is it limits the analysis to the set of genes that are most relevant to the patient's symptoms. So the physician goes to this tool and enters in what the patient has, and then behind the scenes we turn that into a candidate gene list. And so we're using standard ontologies to describe patient symptoms. The current version uses SNOMED CT. We're actually in the middle of migrating over to something called the Human Phenotype Ontology, or HPO. STAGA not only identifies relevant genes, it also ranks them in terms of what we think is most important based on the symptoms. And so currently it looks like this. Enter in a new patient. And we're going to enter in the symptoms for our two patients here: ataxia, developmental disorder, motor function. And with a simple search, now we get back to 133 genes that might be relevant. So remember, we started at over 20,000. Now we're at 133. Ataxia alone gave us 91 genes. Motor delay gave us 37. Muscle atrophy gave us 40, and then smack right in the middle of all of them is this gene called APTX. And so without much foreshadowing, you can see where I'm headed is that this is actually a really important gene to the diagnosis of these patients. So then these patients went into our sequencing center. We did sequencing, we did alignment and variant detection on them. And then the next question is, what does it mean? What does each variant that came out of the pipeline mean? Does it have a functional consequence? And to do that, we use our pipeline called Runes. Runes takes variants, puts them through this set of pipeline stages, and produces a lot of biological information that's basically predicting what the consequence of the variant is going to be. So this is puts it through a combination of in silico tools, some of them that are open source, community tools, some of them that are written by us at CMH. Does comparison to databases. More and more the question is, has anybody else seen this? What did they find out about it? And at the end, we end up assigning essentially a pathogenicity score, a severity score that's based on the American College of Medical Genetics, ACMG. At the end of the Runes pipeline, what we do is we load all of our variants into a database called the Variant Warehouse. And this is keeping track of every nucleotide variant that we've ever seen since we started sequencing 3 years ago. and along with all the characterization information. So as of yesterday, we had 58 million variants in this database, along with all the other characterization info for all these. Technically, under the hood, this is a Java pipeline. It's broken into multiple stages. It runs on the compute cluster. It usually needs, somewhere between 24 to 32 gigs of RAM to run, but it can run on a single node. One of the most important things that I said before is how common is a variant. So within our center, that means, can we identify which samples have each individual variant? And so the way we've done this is with Hadoop and MapReduce. So it sounds like, from talking to Kevin earlier, that in general, this is an audience that knows quite a bit about Hadoop, so I won't go through the canonical word count example with you, except to say that actually, as it turns out, calculating variant frequencies is really similar to that kind of problem. So what we're doing is I kind of sketched out in pseudocode here what our actual map and reduce functions are in our pipeline. So what we do is we have a map function that takes in the sample and the variant file for each patient and basically reads through it and then emits the sample and the variant, which then the reduce, during the shuffle stage, that all gets turned together to where the reduce function gets the variant as a key in a list of all the samples. And then from there, it's just a matter of counting the samples that show the variant. It's a little more complicated than that, but essentially that's what we're doing. So this calculation has to be done every time a new analysis is done because the frequency is going to change every time. So with Hadoop, we were able to do this now in 45 minutes, 10 minutes of which is loading the relational database with the results. And so that's short enough to where we end up doing that every day, multiple times a day sometimes. The inputs into that, we have then, 39 observed variants that in our current patient set. There's 2,700 samples, almost 7,000 individual variant files. That's nearly 2 billion individual variant calls that we're sifting through. So that's about 2 terabytes of input data. And again, we're doing that in 45 minutes. Oops. Okay, so at the end of the runes pipeline, we get something that looks like this. And so I've highlighted the relevant points to this case in yellow. So we can see is categorized as a 1, which is our most severe category. It means this is a known disease mutation. It's very rare. It was only seen in 3 samples, and 2 of them are our patients in the APTX gene. And what it's doing, for those who are biologically on top of it, it's causing a premature stop codon, which means basically this gene's not functioning at all for these patients. Okay, so in the patient's variant set, we've identified that this one is potentially bad news, but for anybody to find it is still an exercise because this is in a variant file that's got 3 million other rows in it. So to answer that question, we've got a tool called VIKING. And VIKING's job is to integrate the output of SAGA and runes and to give a hopefully easy to use interface to the clinicians who are doing the interpreting of genome sequencing results. Technically, VIKING is written as a Java Swing client. It's delivered by Java Web Start. And in its current implementation, it's a client-server app. So the VIKING client is talking directly to the database, the 2 databases, issuing its own SQL. We've been running into some really serious performance issues with its current architecture, basically having to do with dealing with things on a genome scale. It's difficult to hold all of the data for a single patient in memory. Never mind if you want to look at a family, which you often want to do when you're doing diagnosis. So at a minimum, we have about a 6 gig RAM footprint of the client tool, which is a little hard to deploy routinely on workstations at the hospital when even an 8 gig machine is pushing it a little bit. So we're in the middle of re-architecting this to be more of an N-tier architecture. And what we'll do is establish an application server that then the VIKING client will really turn into much more of a thin client that's making RESTful calls to a VIKING server over HTTP and getting something like JSON back. And then that allows us to put a lot of the real heavy lifting on the server, and the server then will take care of merging with the individual databases. One of the key things Viking does is it allows some pretty sophisticated tools for filtering that lets you look through the whole set, and this is done basically using a mixture of strategy and decorator design patterns. So we've got a strategy defined called filter that just says meet if meets filter, and that lets us have concrete implementations that define individual rules for how a variant might meet that filter. So for instance, gene filter, if you give a single gene name, it means give me only the variants that match that gene name. And so then these can be chained together so that you can easily look at just variants that meet all the filters. So this is what the Viking tool looks like, and I went ahead and called up our patient, CMH01. If you look at the patient's symptoms, we got our 3 symptoms entered in. That down below then shows all the diseases and genes that are implicated by those symptoms. If we dial down to say, show us only the most rare variants, and then I've got a few other filters on there. Basically, through using this tool, what we end up getting is a single variant that pops to the top, and this is our premature stop in the APTX gene. So when this works, it really does honestly take 5-10 minutes to go from data results to an answer like this. And I'll be honest and say that it doesn't always work, but it works a lot of the time. So the diagnosis, ataxia, oculomotor apraxia type 1. Both of the patients have homozygous mutations in the APTX gene. The parents were both unaffected carriers. They had 1 copy of the mutation each. The children were unlucky enough to get both of them. What was really neat about this one is that it's actionable. So mutations in the aprotaxin gene cause a CoQ10 deficiency. This is actually a supplement that you can buy in nutrition stores for like a 19-cent pill a day. And it's not a cure again, but it's a way to manage. Kevin, I've run short of my time here, so I'm not sure how I'm doing, but I thought I would give a nod to the audience and talk a little bit about some of the issues that are coming with integrating with the electronic medical record. So clearly, hopefully, my talk has convinced you that there's a lot of interesting, useful information that could come out of your genome. So diagnosis is really just one of them. There's several other applications like very often get called personalized medicine applications where we tailor treatment to your genotype. Some of these things could easily turn into decision support tools that are within the medical record itself. So for instance, if I know that you have a specific version of the CYP2D6 gene, it means that you're not going to metabolize codeine very well at all. And wouldn't that be great if your medical record said that? And if you tried to, maybe if the physician tried to prescribe codeine, we got a prompt or a warning or something like that. So this is going to become more and more common, I think. In fact, it's already happening now. We're already generating genetic information for patients. We don't have a great way of storing it within the medical record at all. And so, of course, to make full use of this stuff, we need to figure out ways to store this in a structured, meaningful way that's scalable, that in order to enable computational analysis and for allow us to build tools that build off this information. So I thought I would show you just quickly, we have one clinical test up and running. All the other tests I mentioned are run on a research basis, but we do have one test that's a clinical test, and this is the way it works. Is currently a physician goes to our Cerner Millennium system and enters an order for a test and just says, I want to order this molecular test. And then separately, they go to our STAT-seq system and enter the patient's symptoms. They go through sequencing analysis, interpretation. Once we've made a diagnosis, hopefully, our clinical director is going to type up a report in an RTF file. And then she's going to upload the RTF file to the medical record system. So unfortunately, that means that there is specific information about what mutations we might have found in that patient, but it's encoded in a file type that's not really going to be easy to do much with in terms of software. And that's a real challenge. So there's a lot of considerations here about when we're starting to talk about how are we going to manage this stuff in the medical record. So the first question to me was, what do you store? So I've said a few times that we're looking at around, say, 3 to 5 million data points or variants for each patient. And I think that's really actually just a starting point. That's just the 5 million data points of the kinds of mutations that we can detect now. There's actually a lot of things that happen in your genome that we're not doing a great job of detecting right now. There's also other different data types that are in your genome that you might want. But let's call it 3 to 5 million per patient. That's the raw data. And then what each of those data points mean, more data. What's the metadata? So for instance, the variant itself is just a position. At this position, I see this change. Doesn't say anything about what gene it is or what the expected function is. So one way to do this might be just store only the things that are the most relevant for each patient. And I guess my immediate question is, well, how do we decide what's relevant? How do you know? What's our process for updating? If we adopted an architecture where maybe a core lab holds the full genome record and the medical record system just initially stores the most relevant, how do we do an update process? How would that work? And then how is this stuff accessed? I think this is a big consideration. So remember that physicians, for the most part, are really struggling with this information and what it means. So no matter what, we've got to put things in a context where they're going to easily understand what they can do with the patient's genomic information. That's not at all straightforward. It also needs to be accessible in real time. So it's not enough just to get it in the system and store it. They've got to be able to access it and query it and get it back out very quickly. And then the final point that I was thinking through that I think is a real challenge is how are we going to manage change? So the human genome sequence actually has gone through several versions. They release new versions all the time. Well, at least every couple of years. And so what that means is if you identify a variant as a position and a change, when the new genome build comes out, that actual position changes. It's a different, you've changed the natural key basically of what the variant is. And so how do you know moving forward, how are you going to update that information? And then the other kinds of change that's going to be really difficult to manage, I think, is the changes in the science and what the meaning of each nucleotide variant is and what we can do with that. And what is the process that we're going to use to address that. If a new piece of science has shown that this variant that was previously thought to be benign is in fact pathogenic, what's the process by which we update the medical record and let patients know? So with that, I'd like to thank you all for coming. I'd like to thank my team, all the folks at Children's Mercy, as well as our supporters. I hope I've given you a little bit of insight into what our world looks like and what some of the challenges for all of us are in the future. With that, I'll close and be glad to take any questions.
Hi. You mentioned a couple things. There are actually like 3 things. There's like the cost, the time it takes, and then the complexity of interpretation. So what I was kind of wondering is, you showed the curve of the cost cost and time changing.
Sure.
If there's going to be like a breakthrough in terms of physicians using this, as common as they do like just regular blood tests or whatever, which front do you think has to be most advanced on, and is the rate of change similar for that as for some of the other ones?
That's an interesting question. Well, one thing I think is to point out that the physicians are actually really eager to start using this kind of data right now, both in terms of research as well as in clinical practice. As inexpensive as genome sequencing has got, and I can give you statistics and say, well, we're able to produce 11 a day. That's still quite a long way from sequencing every single patient that comes into the hospital. But I actually feel very certain that that's where we're headed. So I think that's one inflection point. When can we have machines and instruments that every hospital is able to do this for all their patients? And then the second is a really good question about interpretation and what's it going to take until we can meaningfully interpret all of the data that the sequencers are producing. And I think in some ways we're already there. There's a number of things that can be done, but for the effort to make them happen.
I have a question in relation.
Oh, thanks.
I have a question in relation to the amount of data that's permanently stored. So if I understand it correctly, so you interpret the variants based on a reference genome, and that reference genome is common, through a bunch of ones that you basically created those variants off of. What is the benefit of storing the pre-variant data rather than just storing those different versions of the reference genome where you could work backwards if you needed to?
Sure, that's a good question. So you could look at it as, why don't we just store basically the variant list as a kind of delta compression, and that's it. And the reason why we store all of the data that leads to those variant calls is because there are always developments in ways that we can interpret that data to lead to new variant calls, to lead to new types of things that we haven't actually thought of yet. There's certain mutation types that are kind of buried in there that the software has a hard time calling right now. And also because at the current state, that ends up being important secondary data that our clinicians look at to try to determine if they have something real on their hands or if it's maybe an artifact. So it is actually useful in the interpretation to look at it's not exactly raw data, but let's call it intermediate data, and look at the read alignments and see how the variant call was made.
So I have a question about, in your beginning slides, you have slide mentioned that the reads is really short and the error rate is really high. I was wondering if that error rate is talking about the after the assembly, or what was that high error rate?
Okay, it means in the actual sequencing, there's a fairly high error rate for calling the wrong base. So if you have a short read of, say, 125 base pairs, I think it's 1 in 1,000 bases are going to be incorrect, meaning maybe the machine called a C when in fact it's a G. That's what I mean by error rate. And so a lot of the different sequencing vendors all have slightly different error profiles that are characteristic of their data. Illumina's happens to be towards the end of the variant, of the read. Quality drops off. That was a question. Yeah, I mean, the way we address that is by doing that tiling where we're measuring basically every base in the genome at least 30 times. And so that's why variant calling is a consensus over the multiple reads. But that's in comparison with Sanger sequencing, which is still considered the gold standard of sequencing. Very high fidelity, 1,000 base pair reads. Yes?
Is the alignment process parallelized by individual chromosome, or do you parallelize it on a different basis?
It can't be parallelized by chromosome because you don't know where each read is yet. So what you're doing is you're mapping it, and what we tend to do is basically on a file basis. There's a few different ways that you can take the input set and split it up over the whole cluster. It just happens that the sequencing instrument produces many different data files that basically for us is an easy unit that we use for each compute job.
So the FASTQ files contain sequences that are basically agnostic of the chromosome itself, but there are references for individual chromosomes, correct?
Yes, absolutely. Right. So, okay, the very first thing that happens, the sequencer produces this giant bag of little 100 base pair reads that you don't know where they go. And so the alignment process maps them to the reference genome. Once that's done, then you're absolutely right. There's a lot of processes that can be done, can be parallelized based on position, because there are still things that can be done independently of each other. So, for instance, variant detection can be done in parallel. You can look at 1 million base pairs on chromosome 1 and do all your variant detection within that. At the same time, you're doing variant detection on a completely different part of the genome. That's exactly what we do.
Is the industry as a whole primarily doing this for just children, or is also sequencing adult genomes and things like that?
Absolutely. In fact, I might say that we're somewhat unique in that we're a pediatric focus. So for instance, cancer is a very large topic in sequencing. It makes a lot of sense. There's almost zero work going on in pediatric cancer, or there's not anywhere near what's going on in adult cancers. And of course, I should mention that we shouldn't be species-centric, that there's lots and lots of sequencing and genomics that's going on that's not human sequencing. It just happens that we tend to be biased and focus on that. But there's lots of great work going on in the plant world, and lots of applications there in the agriculture world that's going on with this technology has been similar, disruptive.
You mentioned that your clinicians are readily hello. You mentioned that your clinicians are very eager to get the data, and are they helping shape your potential next assay that's going up in terms of the genetic loci you may be focused on?
Yes. Although what we're doing right now, we spent the first few years focused just on the problem of diagnosis, and that fit very well with our existing molecular genetics department. And now we have that up and running, and what we're starting to do is work with other departments within the hospital to answer their specific needs. So one of the very first ones is cancer, and can we come up with a directed panel? There's a lot of work that's been done to show that your individual genotype has a big effect on how you might respond to treatments, and so our oncologists are really eager to do that. We've also got some interesting projects starting up with the neurology department, looking at basically sequencing brain tumors and looking at how those happen. Okay, well, looks like we might be at an end. Thank you again very much.